Okay, our last video is the coolest part. It's the part with the most steps. And this is something we haven't talked about before. It's called meiosis. M-E-I-O-S-I-S. Maybe I'll make it pretty. This makes gametes. Gametes are also called sex cells. These sex cells are haploid. They need to have half the DNA so that they can go meet up with another cell that has half the DNA and make a new organism. We'll talk about that tomorrow. So half the DNA of a somatic cell. If you didn't do this, then a human would have 46 chromosomes, and then if you just sent your 46 and 46 came from the other parent, they'd have twice that many. You'd end up with tons of chromosomes. That would not work. So this is still Fred. He still starts out with a cell that looks like all the other cells in his body. Big, medium, small. Big, medium, small. And he goes through S phase. So this part looks the same. S phase. Now I have 6 duplicated chromosomes. Oops, ignore that. And in this case, some really cool and weird stuff happens. So before they divide, my homologous chromosomes are going to hang out right next to each other. So here's my chromosome 1 hanging out right next to chromosome 1. Chromosome 2 is going to hang out right next to chromosome 2. They don't necessarily line up. It's not that red is always on the right. In fact, let's say that blue happens to be on the left over here. So they line up right next to each other. These are called tetrads. So when they form pairs of homologous chromosomes. They actually wrap their arms all around each other and they swap pieces. So remember we saw earlier that there's such a thing called recombination? This is when it happens. Oops, not an S. This is when it happens. Is that they'll actually swap pieces. So now this is when the drawing gets complicated. Now I have this chromosome, this one, and I'll put that there because these ones swapped, maybe they just swapped one end of their DNA. And now this little guy has a red one. And over here, the same thing happened. Let's see, oops, medium, and super small. Oh, I'll fix that. So now I have some pieces that look kind of new. And instead of pulling apart these identical pieces, what happens in the first division is that these homologous chromosomes get pulled apart. My phone is getting emails. So my homologous chromosomes split, and I end up with, don't make your drawing too pretty or it'll take you too long. I'm doing it by color. The big one, medium one, the big one should be bigger, sorry. And the small one. Alright, so I'm now split them in half. This is called meiosis 1, the first division. Then the second division looks a lot like mitosis. The chromosomes that are left in each cell line up so that these almost identical now, but they're not completely identical because some of them swapped pieces. They line up so that they can get pulled separate parts of the cell. This has a little N. big, medium, small. Medium should be bigger. And then they will get divided in half. So they'll split again. This piece will go this way. This piece will go that way. This piece will go this way. This piece will go that way. They'll split in half. I'm not going to draw that on both sides. And I will end up with two cells that came from this one. So I've got this piece on this left side, and then I have this piece, and then I have this piece. And none of these have any weird spots because they didn't exchange any pieces on the left side. If I looked at the left side, everything looks normal. And if I look on the right side, those are where all of my recombination happened. So I've got 1, 2, 3. So this one split into two cells. I'll keep adding my arrows. And then over here I do the same thing. I look at what I've got going on. All right, everything on the left side, I split these identical pieces of chromosomes in half so they're not duplicated anymore. This is my big chromosome piece, my medium chromosome piece, and my small chromosome piece. Then over here, if I look again, big piece, medium piece, and the small piece. So now I have gone through, this is called meiosis 2. So there's two divisions, meiosis 1 and meiosis 2. And I end up with 4 daughter cells. The question is, do they look like my parent cell? They look similar, but there's definitely a difference. First of all, my 4 daughter cells, does any one of them look exactly the same as the other? All are unique. There's no one that looks like the other. So I get 4 different cells, which is cool. Secondly, let's go through and do the same thing that we did before. I'm going to ask the question, haploid or diploid? Let's scoot that over. The question is haploid or diploid? And then, are they duplicated? Or, okay, my phone kept running out of space. So, I took a few notes that you didn't see. I remember we're asking, is it a haploid or diploid? Is it duplicated or unduplicated? In the process, I've somehow misplaced my green pen, so this is going to be in blue. We reminded ourselves that for Fred, N, the number of chromosomes that come in a set, is 3. And that 2N for Fred is going to be the diploid number, is 6. Sorry, I changed colors. Let's do this in orange. So number 1, how many chromosomes? Do I have 3 or 6? I have 6. So I can call this diploid. By the way, anytime in this video, if you want to try yourself, I'm going to go through all of these and label them haploid or diploid. You can try and then check your answer. 2 is duplicated or unduplicated. I can see that they're not X's, they're just lines. They're unduplicated. This one, same questions. I got 6 chromosomes, diploid. I have, now they're X's, so they've been duplicated. Part 3, it's the same cell, I'm just moving stuff around. So I've got still diploid, and it's duplicated. All right, here, let's check this out. So assuming that these are already 2 different cells, one is, is it diploid or haploid? Is it duplicated or unduplicated? So we'll do number 2 first. Each one of these is still stuck together. So if I look in this one cell, I can see that my chromosomes are duplicated. Even though now they're not exactly the same. I've done some weird stuff. So I can definitely say they're duplicated. But then let's look at one cell. So how many chromosomes in this one cell? If you want to try counting them, go ahead. I've got 1, 2, 3. Over here I've got 1, 2, 3. And I look up here, 3 is N. I'm now down to half the number of chromosomes because I split. On this stage, I split homologous chromosomes. Split homologous chromosomes. I'm not diploid anymore. I'm now haploid. So meiosis 1 takes my cells from being diploid all the way to haploid. They're still duplicated, but they're these duplicated haploid cells. You can even label them that way. Duplicated haploid. Here, we'll skip this step because it's not clear whether I mean before or after they actually divide, but let's look down here. 1. Oh, you have something written here. We'll put that here. Are they duplicated or unduplicated? Are they haploid or diploid? So in each one I can see there are only 3 chromosomes, which makes them haploid. And they're unduplicated. I pulled the duplicated chromosomes apart. So I went from, at the beginning of this, once I went through S phase, I had diploid duplicated cells. And now I have haploid unduplicated cells. So I've gone from 1 cell to 4 cells, and it doesn't look identical to the parent. It doesn't look identical to anything. I have 4 different cells. They're all called gametes. So maybe I made 4 sperm cells. They're gametes, or sex cells. And they're going to go off and they're going to meet up with another cell, and they're going to make a child or a new Fred, or whatever it is we're talking about. So these are the end of your notes. Go online and take the exit ticket that it says here on your paper. It doesn't say it on mine. When you're done with all of those things, you're going to just begin doing these pages. Finishing this packet is your homework.